Good morning, everyone. This is the Orlando GI Journal Club. This is May 7th, 2025. Welcome, everybody. We're going to do something a little bit different today. We all actually just returned from DDW. So I've picked out some of the best abstracts presented at DDW this year. And so we'll review some of these abstracts, which is, some of the most cutting edge research going on worldwide. This is a study looking at the safety and efficacy of inebilizumab in IgG4-related disease in participants from pancreatic and biliary involvement. This is a phase 3 trial. Inebilizumab, which we're going to shorten to INEB, thank goodness, is an anti-CD19 antibody that results in rapid and durable B-cell depletion. These authors conducted an international randomized, blinded, placebo-controlled phase 3 trial to look at the safety and efficacy of INEB as treatment for IgG4-related pancreatic and biliary diseases. The participants were enrolled if they had a history of at least 2 organ involvement and IgG4-related disease flare that required glucocorticoid treatment. Patients were randomized to INEB or placebo and were treated on days 1, 15, and week 26 of a 1-year randomized control period. Steroids were tapered to discontinue at the end of randomized control period, week 8, and other immunosuppressive therapy was prohibited during this time. 135 participants were enrolled. 52% of these patients had pancreatic, and 32% of these patients had bile duct involvement. INEB reduced flare risk, which was the primary endpoint, compared to placebo in the pancreas group. And there were no flares in the bile duct or combination of pancreas and bile duct group, compared to 12 and 11 respectively in the placebo group. Proportion of participants achieving flare-free, treatment-free remission were higher in INEB versus placebo, with odds ratio of 10.8, 35.8, and 66.4 for pancreas, biliary, and combination groups, respectively. Steroid use was reduced in INEB and serious adverse events, and INEB were mainly hyponatremia and fibroadenomatoid mastopathy. No mortality was observed in any of the study participants. In conclusion, CD4/19 targeted B-cell therapy is B-cell depletion therapy is effective and safe in pancreatic and biliary IgG4-related disease. Our next study looks at the yield of interval fecal immunochemical testing following colonoscopy. This is a large nationwide study from the VA database by Wilson et al. Current guidelines advise against FIT testing within 10 years of a negative colonoscopy, yet it is often performed prior to the next recommended screening, and currently there are no data on the outcomes of interval FIT testing. Data obtained from the VA system. Adults who underwent colonoscopy followed by FIT within 10 years were included. Polyps on index colonoscopy prior to FIT and post-FIT colonoscopy were identified. Advanced adenoma was defined as adenoma with high-grade dysplasia, tubular villus adenoma, or cancer. Primary outcomes were prevalence of interval FIT testing, predictors of positive interval FIT testing, and findings on follow-up colonoscopy. 4,700,000 individuals underwent colonoscopy. And among these patients, 10.9% had an interval FIT testing, of which 15.6%, that's 82,000 patients, was positive. Index colonoscopy was negative in 59% of IFIT positive versus 65% of IFIT negative patients. Of the 82,000 patients with positive interval FIT testing, 49.9% underwent follow colonoscopy, with advanced adenoma detected in 8.6% and colorectal cancer in 3.2%. Independent predictors of advanced adenoma in these patients who had positive interval FIT testing were male sex, those over the age of 50, and those with advanced adenomas on index colonoscopy. So in conclusion, prevalence of interval FIT testing was high, but this was positive in 15.6%, with advanced adenomas detected in 8.6% and colorectal cancer in 3.2%. And therefore, it looks like the yield of interval FIT testing is high, regardless of the recommended guidelines. Our next study is a multicenter, double-blind, randomized, placebo-controlled trial to evaluate the efficacy and safety of eloxapitat in treating patients with chronic constipation. The study was to evaluate the efficacy and safety of eloxapitat, which is an ileal bile acid transporter inhibitor, in treating patients with chronic constipation. In this multicenter randomized double-blind placebo-controlled study, comparing treatment with 12 weeks of eloxapitat versus placebo in patients with chronic constipation based on Rome 4 criteria. Patients were randomized in a 2-to-1 ratio to eloxapitat or placebo. Primary endpoint was complete spontaneous bowel movement rate during 12 weeks of treatment, and additional 4 weeks of follow-up was applied to observe withdrawal and or rebound phenomenon. 361 patients were randomized to eloxapitat or placebo. The overall complete spontaneous bowel movement response rate during the 12 weeks of treatment was significantly higher in eloxapitat versus placebo, and at each week of treatment, but not at the last visit of the follow-up period. The mean stool consistency scores were higher in eloxapitat group in each treatment week, but not during follow-up period, and there were no significant differences in safety assessments. Eloxapitat could significantly improve bowel function and reduce severity of symptoms in chronic constipation compared to placebo. Next, we're moving on to interventional endoscopy studies. This is the results of the SHARP trial, which is looking at minor papilla sphincterotomy in patients with acute recurrent pancreatitis and pancreas divisum. Sham-controlled trial to measure the impact of minor papilla endoscopic sphincterotomy on the risk of subsequent acute pancreatitis was conducted. This was a sham-controlled, single-blind, multicenter randomized trial, and they included patients with history of at least 2 documented episodes of idiopathic acute pancreatitis, at least 1 within 24 months prior to randomization, and pancreas divisum confirmed by MRCP. Patients were randomized to EUS with sham ERCP, where the pancreatic stent was placed into the duodenal lumen, or with minor papilla endoscopic sphincterotomy and placement of a prophylactic pancreatic stent. The primary outcome was development of acute pancreatitis, defined as a time to event measure. Patients were followed up for a minimum of 6 and up to 48 months. 148 patients were randomized, 75 patients to minor papilla endoscopic sphincterotomy, and 73 patients to sham ERCP. Subjects had a median of 3 acute pancreatitis episodes prior to randomization, and the median age at first acute pancreatitis episode was 51 years. During median follow-up of 33.5 months, 35% of the patients in the minor sphincterotomy arm had an acute pancreatitis event, compared to 43.8% in the sham and the adjusted hazard ratio was 0.83. That's with a 95% confidence interval spanning 1, and therefore there was no significant difference between these 2 groups in the occurrence of acute pancreatitis. The incidence rate ratio was 0.23. 0 for minor sphincterotomy versus 0.277 for sham. And this means that acute pancreatitis episode frequency after randomization was reduced in both study arms, and there was no difference in incidence of chronic pancreatitis, diabetes, and exocrine pancreatic insufficiency during follow-up. Therefore, it appears that a minor papillary sphincterotomy does not reduce the risk of acute pancreatitis or related sequelae in patients with acute recurrent pancreatitis and pancreativism. Next is a randomized trial comparing EUS-guided choledochondrogestomy and ERCP for malignant distal biliary obstruction. The study was conducted to compare the outcomes of EUS-guided choledochondrogestomy with ERCP when used as the primary modality for malignant biliary obstruction. This is a multi-center randomized trial from centers in Italy of patients who had obstructive jaundice due to malignant distal biliary obstruction, as long as their common bile duct was at least 15 millimeters. Patients randomized to either EUS-guided choledochondrogestomy or ERCP. The post-procedural acute pancreatitis rate was the primary outcome. Actually, the interesting thing is that I was in this, I listened to this presentation live, and the primary outcome measure actually had changed on the final presentation compared to the abstract, which I thought was interesting. Anyway, so the enrolled 220 patients, 111 to EUS-guided choledochondrogestomy, 109 to ERCP. EUS-guided choledochondrogestomy group showed a significant lower risk of post-procedural acute pancreatitis at 1.8% versus 7.3% in the ERCP group. Technical success was significantly lower for ERCP at 17.9% and 94.6% in the EUS-guided choledochondrogestomy group. And the mean procedural time was also significantly shorter for choledochondrogestomy compared to ERCP. They found no difference in adverse events, clinical success rate, stain patency, or overall mortality. In conclusion, the authors concluded that EUS-guided choledochondrogestomy superior to ERCP in reducing post-procedural acute pancreatitis risk, and also higher technical success and comparable clinical efficacy. And these results support the use of EUS-guided choledochondrogestomy as a primary approach in patients with dilated common bile duct. I think this study has a lot of good points in that it's a multi-center randomized trial with a large number of patients enrolled. I think what really worried me while listening to this presentation at DDW was that the technical success of ERCP is very low in this study. It's only 78.9%, which is considered low for ERCP. And when we actually asked the presenting author why the ERCP rate was so low, the answer they gave was that the ERCP technical success rate of ERCP was low in their study because they didn't really try very hard with ERCP, because the option was that if ERCP failed, they would cross over the patient to EUS-guided choledochondrogestomy, which to me was extremely alarming because it obviously introduces a lot of bias towards superiority for EUS-guided choledochondrogestomy. So I think this is a good study in that obviously it adds to the literature, and obviously they had a good result with EUS-guided choledochondrogestomy, but there's also been 5 other randomized trials, including one by a group who we first published in 2018, should be kind of interpreted with caution. Next is a randomized trial of endoscopic transpapillary gallbladder stenting versus EUS-guided gallbladder drainage in high surgical risk patients with acute cholecystitis. This is a study from Thailand. The aim of the study was to compare the long-term effectiveness of endoscopic transpapillary gallbladder stenting, so doing an ERCP and inserting stents through the cystic duct into the gallbladder, versus EUS-guided transperineal stenting of the gallbladder in patients with acute cholecystitis. High risk surgical patients with acute cholecystitis were randomized to endoscopic transpapillary gallbladder stenting and EUS-guided gallbladder drainage. For the transpapillary stenting group, 7 French by 15 centimeter double pigtail plastic stents were used. And for US-guided gallbladder drainage, they used either aluminum piece metal stents or fully covered metal stents with plastic stents for anchoring. The primary outcome measure was the recurrence of acute cholecystitis during follow-up. 80 patients were randomized to either transpapillary stenting versus US-guided gallbladder drainage. Transpapillary stenting had a lower success rate, technical success rate, in the US-guided gallbladder drainage at only 88%, with 100% technical success rate for US-guided gallbladder drainage, with similar clinical success. Transpapillary stenting was unsuccessful due to an acute cystic duct angle in 3 patients and cystic duct stone presence in 2 patients. There was no difference in procedure-related adverse events between the 2 groups. The rate of recurrent acute cholecystitis was higher in transpapillary stenting than gallbladder drainage. The Kaplan-Meier analysis revealed a significant higher rate of recurrent acute cholecystitis in transpapillary compared to US-guided gallbladder drainage after median follow-up of 383 days. In high-risk surgical patients with acute cholecystitis at 1 year, US-guided gallbladder drainage provided significantly lower rate of recurrence of acute cholecystitis and higher technical success than transpapillary gallbladder drainage. This is a study that we presented at DDW. This is comparing US-guided gastroenterostomy for malignant gastric outlet obstruction versus surgical gastrojejunostomy. We called it the GOOSE trial. Surgical gastrostomy is the standard method for palliation of malignant gastric obstruction, but US-guided gastroenterostomy is being proposed as a novel alternative approach. We compared both methods to assess whether the clinical outcomes of US-guided gastroenterostomy is superior to surgery. We performed a international multi-center randomized trial with 6 centers from 3 countries, United States, India, and Germany. We enrolled patients with malignant gastric outlet obstruction due to unresectable cancer with GOOS symptom score of 1 or 0. That means their maximal oral intake is liquid only. Patients were randomized in a 1-to-1 ratio to receive either US-guided gastroenterostomy or surgical gastrojejunostomy. And we had a composite endpoint of either inability to tolerate a solid diet prior to hospital discharge and or need for reinterventions, or supplemental nutrition and procedure-related adverse events. 74 patients with malignant gastric outlet obstruction were randomized to either US-guided gastroenterostomy or surgical gastrojejunostomy. When looking at the primary endpoint, again, of either not being able to tolerate a solid diet at hospital discharge, need for reinterventions, or supplemental nutrition and procedure-related adverse events, this occurred more commonly in US-guided in surgical group at 38.9% versus the US group at 7.9%. Also, more patients in the surgical gastrojejunostomy group were not able to tolerate solid diet at discharge, required reinterventions or supplemental nutrition, and there was no significant difference in adverse events between the 2 groups. Additionally, when looking at the other secondary outcome measures, which I have included in the slides here because we have because they are quite extensive, we also found better quality of life for EUS, lower costs, and more rapid initiation of chemotherapy. So in patients with malignant gastric outlet obstruction, US-guided gastroenterostomy was superior to surgical approach, as fewer patients had difficulty in tolerating an oral diet. of solid food to hospital discharge, and also required less reinterventions and lower and less need for supplemental nutrition. Next is a study looking at EOS-guided liver biopsy and portal pressure gradient measurement compared to transjugular approach. This was a randomized trial looking at the performance of EOS and transjugular approaches for consistent portal pressure gradient measurement and quality of liver biopsy. Participants were randomly assigned EOS guided or transjugular route for portal pressure gradient measurement and liver biopsy. And the primary endpoint was a proportion of high-quality liver biopsy specimen, defined as specimens that were at least 25 millimeters in length, with at least 11 portal tracts and consistent portal pressure gradient readings, with a duplicate or repeat measures within 1 millimeters of mercury of each other. 58 patients were randomized, 29 to EOS and 29 to transjugular route. The primary endpoint was met in 24 EOS patients and 12 transjugular patients. That's 82.8% versus 41%, which was a statistically significant difference. The mean procedure duration was 20.7 minutes for EOS, 48.8 minutes for transjugular route. The mean total aggregate length of the specimens for EOS was 117 millimeters versus 29 millimeters for transjugular route, and the complete portal tracts per specimen was 29 versus 11 for EOS and transjugular routes, respectively. Consistent portal pressure gradient measurements were obtained in 89.7% for EOS, 58.6% for transjugular route. Therefore, EOS is superior to transjugular route for the simultaneous acquisition of high-quality liver biopsy specimen and more consistent portal pressure gradient measurements. Next is interim results from the SGENOVA trial. This is a phase 2A randomized placebo-controlled double-blind study to assess the safety of pharmacokinetics and pharmacodynamics of AGMB-129 in patients with fibrostenotic Crohn's disease. AGMB-129 is an oral GI-restricted activin receptor-like kinase 5 small molecule inhibitor developed for treatment of fibrostenotic Crohn's disease. It bypasses clinically relevant systemic exposure via high first-pass liver metabolism to avoid toxicities reported in prior activin receptor-like kinase 5 inhibitors. Patients with symptomatic fibrostenotic Crohn's disease were randomized in a 1:1:1 ratio to receive either the AGMB-129 molecule, a 200 milligram dose BID, or the IGMB-129 drug, 100 milligrams once daily, or placebo for 12 weeks. The primary objective was safety and tolerability of AGMB-129. Secondary objectives were to evaluate plasma levels and changes in gene expression in ileal biopsies collected at screening and week 12. Data on 41 patients with mean disease duration of 18 years and 73% on concomitant biologics were presented. Treatment with AGMB-129 was safe and well tolerated with no cardiac toxicity or other adverse events. Low systemic exposure to AGMB-129, but high exposure to its ALK5 inactive main metabolites was noted. Mucosal ileal biopsies revealed downregulation of fibrotic inflammatory gene sets with improved symptom severity and disease activity. So treatment with AGMB-129 was safe and well tolerated with significant downregulation of both the inflammatory and fibrotic pathways and trended improvement in symptom and endoscopic outcome measures, and therefore AGMB is a promising novel treatment and further evaluation is needed. Due to the time, I'm going to skip over the next two abstracts, and I'm just abstracts. What I would recommend is if you come to on Saturday at 5:30 p.m. Eastern Standard Time, we will be doing a DDW update live on online, and we'll be presenting lots and lots of really good abstracts from DDW. It's always a really good session. I beg your pardon, at 6 p.m. Eastern Standard Time. I have to be there at 5:30. I don't know what time I told you, but everyone else can be there at 6. I apologize. So 6 p.m. Eastern Standard Time, we're going to be doing a DDW update presentation online. Please register and join us. Thank you so much. For our next monthly GI Journal Club, we'll be presenting on June 4th, 2025. Thank you so much. Please have a wonderful day, week and month, and look forward to seeing everyone next month. Thank you so much. Bye.